Next plenary session, our next plenary talk, and it's going to be taken by Professor Kemi Amodu, but I have to call on the microphone now Dr. Miriam Oloye to please give us a citation of our next plenary speaker. Professor Kemi, you can please come up stage.
My Dean, Professor Nkiko, let me align myself with the protocol, existing protocol. I'm reading before you the citation of Professor Olukemi K. Amodu. Professor Olukemi Kehinde Amodu is a distinguished scholar holding the prestigious position of Professor in Genetics, Molecular Biology, and Public Health at the esteemed Institute of Child Health, College of Medicine, University of Ibadan, Nigeria. She's also the director of the Center for Molecular Bioscience and Medical Genomics, University of Medical Science, Ondo. Her academic journey includes the attainment of a master's and doctorate in molecular biology and genetics from the University of Ibadan, followed by postdoctoral training at Harvard University in Cambridge, USA. Throughout her career, Professor Amodu has actively contributed to various public health genetics research initiatives focused on childhood infectious diseases and non-communicable diseases. Our primary expertise lies in human genetics, genomics, delving into the molecular mechanisms underlying disease pathogenesis, particularly in infectious diseases and more recently, non-communicable diseases. Please, let's share her up. Let's give her applaud her. Her pioneering work in exploring human and parasite genetics, variability in malaria pathogenesis, has yielded groundbreaking insights, fostering collaborations with both local and international experts, including significant involvement with esteemed consortia such as the Malaria Genetics Epidemiology Network, Malaria GEN, and EU-funded networks. Her scholarly contributions are substantial, encompassing over 120 publications, comprising books, articles, data sequence databases, and patents with a Google citation count of 3,142. Achieving tenure at the University of Ibadan marked a pivotal moment in her career as she dedicated herself to establishing a genetics molecular biology laboratory from scratch at the Institute of Child Health. A central goal for Professor Amodu is to enhance the capacity for research and application of genomics, bioinformatics, biotechnology, and genetics molecular sciences in addressing infectious and non-communicable diseases of public health significance in Nigeria. Our efforts to integrate basic and clinical research into translational studies aim to elevate patent management strategies. Demonstrating her commitment to education and skill development, she spearheaded the creation of a groundbreaking academic master's and PhD program in public health biotechnology at the Institute of Child Health. This innovative curriculum intertwines essential aspects of biology, genetics, genomics, molecular biology, bioinformatics, biotechnology, epidemiology, and public health. The success of this program is evident through the graduation of numerous students across various levels, with Professor Amodu supervising several PhD, MPhil, MPH, and MSc students. Furthermore, our leadership extends to driving initiatives for technological advancement in education, notably establishing the first hybrid electronics classroom at the University of Ibadan, in partnership with the First City Monument Bank PLC, FCMB, fostering an environment conducive to borderless learning. Since ascending to the position of professor at the Institute of Child Health, her influence has been significant, serving as acting as substantive director of the Institute at different times. Noteworthy contributions include a role as the principal investigator for the Bill and Melinda Gates Grand Challenge Exploration Grants, focused on devising innovative strategies to enhance vaccination demand among working mothers in the Ibadan metropolis. Additionally, she remains an active member of the American Society for Human Genetics, contributes as an expert reviewer for granting bodies including the EDTCP3, and holds the esteemed position of Deputy Editor-in-Chief of the Nigerian Journal of Child and Adolescent Health. Ladies and gentlemen, let's welcome Professor Amodu as she delivers a lecture. You're welcome, ma'am.
Can I lift it up here? Good afternoon, everybody. Please permit me to stand on existing protocol. You're all welcome here. Thank you very much for taking the time to listen. The title of my presentation is Genomic Insights and their application in modern healthcare. Okay, so I'll run through this. This is my outline. I'm going to run through it. We'll talk about the genome and what the application of genomics is in healthcare and what the future direction. So we're starting off with introducing what the genome is. And the genome is a complete set of genetic information that is encoded in our DNA. That means the complete set of genes in any organism. So I have a total number of genes, and if you are going to deal with me, you talk about my own genome. So that's what a genome is. And it differs from different organisms, from different species. For the humans, we have about 3 billion pairs of DNA, and they are organized into 23 pairs of chromosomes. From our basic biology, we know we have 22 pairs of autosomes, and we have 1 pair of sex chromosomes. For the female, you have the XX, and for the male, you have the XY. So that is simple biology. We are going back to simple biology. And all genes are important for development, for functioning, and reproduction. And interestingly, we are still doing a lot of research to know the function of some genes. So with the Human Genome Project, which I'll still talk about later, we've been able to discover a lot of genes, and scientists have been able to, they're still continuing the work to understand the function of some certain genes. Now, we need to understand the human genome. This is crucial, because when we understand our genetic traits and how it affects our diseases, and it will help us to be able to know how to, drive healthcare. The previous speaker, though I didn't stay around much, talked about using synthetic, developing drugs. And now we are talking about there's a side developing drugs, and you cannot develop drugs in isolation or silos. We must now begin to think of how the ones developing drugs, we also work with people who are looking at the genetic traits so that we will see what are the important things. As we go on, I will explain a little better why this is important. Okay, so genomics, I'm sure you've heard about omics. There's so many omics all over the place, but genomics was the first omics. And because of the rise in innovations, development in molecular biology, it started a drive in the 1990s. I will go through that developmental stage. And that was when the Human Genome Project started. It was about 1990. And they started the work, little by little. Eventually, they were able to get the work done by 2000, and up to a point at 2004. And I can tell you that since then, we now call that the post-genomic era, meaning that after we discovered the human genome, the era coming behind it, there has been a lot of discovery since then. It's a very interesting time for us if we will just key into it. So genomics is the study of the entirety of the organism's genome, how we interrogate the different variations in our genes, and how these genes contribute to what we call Mendelian diseases and other complex disorders. So I'm giving an example of the other omics. I'm just going to mention them in case you come across them so that you don't just say, which one is this omics again? You understand what they are all about. So we have proteomics, that is the study of proteins. Then we have the metabolites, metabolomics. So metabolites are also important in this era. Then we have the environmental DNA, which is metagenomics. That means from the environment, you can track diseases. That happened a lot during COVID. In many developed countries, they were able to track, the measure of the COVID, even with the water flowing out of systems. So there's a lot about these times that we are in. And now we have something we call phenomics. Phenomics is now important because phenomics is about phenotypes. We need to be able to classify what particular phenotypes we see. So if a person in a population now, the phenomics may be different. People that have maybe type 2 diabetes in Nigeria may bring out a completely different phenotype from maybe the African American or Europe. So we need to be able to classify and categorize our phenotypes. So that is why phenomics is very important now. So what are the important developments in genomics? This slide is busy, and I'm not sure how we are going to be able to look at it, but if you can see that it started from 1990, just as I mentioned. That was when the Human Genome Project started. And there have been a lot of challenges along the path. Eventually in 2003, they completed, I would say at that time, what they knew about the Human Genome Project. And then afterwards, we had something called the first HapMap report. In fact, they came to Nigeria and they used the Yoruba population. And that project is so important today. Why is it important? Now, when we see, when we type different genes, we're able to compare it with this HapMap project and see, what are the minor allelic frequencies? Why are those things necessary? Those things are necessary because, for example, if you have a particular gene that may be associated with a disease in a population, by doing the genotyping, you'll be able to know whether that gene is actually prevalent in our population, maybe to a large degree, and then you'll be able to see which one is actually the at-risk genotype. For example, we all know about the HBAA, HBAS. Please, we all know that the S is the risk one, right? That is actually the phenotype. We know that the S is the risk. So that is why we need to do all these projects. So in 2005, we had the first HapMap. Then we had 1,000 genome projects. And then in 2013, there's a gene editing tool that was discovered. I will still talk about that a little more, but I'm not going to bore you with too many technical details. So by 2022, the entirety of the human genome sequence was completed. So even though a part was completed in 2013, the full thing was now completed in 2022. So this HapMap, like I mentioned, they were landmark projects, and they help us to understand more about our genes and how they vary from one person to the other, or how should I put it, how the genes affect diseases. How, if you have a certain variation in a gene, it may make you more susceptible to a disease or not. So this is just the example of the HapMap project, the 1000 genome. So from there, it's a catalog of what you call single nucleotide polymorphisms. These are different variations in our genes. So with the HapMap project, there's a haplotype map, and it has common patterns for human genetic variation, and it's a very useful tool for those working in that field. And then on top of it, we now have the 1000 genome project that identified about 84 million SNPs and different variations in the human genome. So what are the tools for what are the genomic tools and technology that we have been able to see in these last few years? Again, there's a map showing that a study for genomics even started in the 17th century. There's a report of somebody observing an oral microbiota, even from back then. But then study started slowly, little by little, and it seems as if in the 20th, 21st century, everything just boom, and there's more knowledge available. Now we have a lot of studies going on. We can talk about so many genomics, and these genomics these omics come about because we were able to develop many tools. For example, we just had the Sanger sequencing to start with. Then afterwards, biotechnology developed, and people began to develop different sequencers, and we now have varieties of sequencers that we can use. So now we are sitting in a time where we can answer our problems, and I just want to challenge us here in Nigeria. There's still a lot of work to be done. We can sit down, I can do the entirety of this presentation and talk about what people have done, but I will keep interjecting on the things that I think we should start paying attention to, because that is important. I'm not just here to just give a lecture and tell you all the wonderful things that are happening in the genomics world, and we can do that and close shop and go home. But I think we can start, thinking about the little things we can start doing to start building. They started from somewhere after all, and we should not be in a hurry. We should start off from somewhere so that we have extreme data that is useful to us here in Nigeria. All right, so we have these different genomic tools. We have the PCR. It's expensive, but I think I've given a talk here before that I mentioned that, yes, these things are expensive, but we can start, little by little. We can combine with each other and start, answering some questions little by little. So there's the genomic. So we are here. There are other tools available. There are SNP analysis, genotype arrays. And by collaborating with people, we can start using these tools for ourselves. So we can do a lot of things, even with the little that we have. And I just want to encourage us that we shouldn't just think that it's impossible. Nobody is going to type our population the way we can type it. All right, so we have next generation sequencing now, and that is high throughput. We can do a lot of whole genome sequencing. We can do genome-wide association studies. We can partner with different collaborators. The interesting thing about combining with other partners is that, yes, they use your data for the large pool, but you can retrieve your data and use it to apply it to your own population. I was looking at some of the data I'd collaborated with a large consortium with, and I realized that there was a paper that I did we're doing phenotyping studies, and we're looking at how sickle cell traits, that is AS, and ABO blood group is associated with severe malaria. And our findings show that children that have group O blood group, they protect against severe malaria. But children with blood B group, are more susceptible to severe malaria. And then I now saw that in the general large data, they actually confirmed it with their genome-wide association studies. So now I can authoritatively say that children, if you have a child that has B blood group, that child should be watched even more carefully than other children, because that child can come down more with malaria. So, those are ways we can answer our own problems and, share our data here. Okay, so there are other tools, the CRISPR-Cas9, and then the bioinformatic tools. I want us to encourage young people around who are into computer studies. We can do a lot with bioinformatics, but we don't have as many people who are interested in bioinformatics, and we need that particular field to be able to answer so many questions. So I'm throwing a challenge to even the younger people. Get involved. You can play a role. All right, so what are the applications of genomics in modern healthcare? We've heard about precision science, precision medicine. Disease prediction is one of it, and prevention. Pharmacogenomics, genomic testing and screening, gene therapy. How do we manage infectious diseases and reproductive health? So and they're doing a lot of this even with reproductive health. We have many clinics now that do, I've forgotten, what's the name? They type the mother, they do a lot of genomic testing now before, please remind me, ma, a woman wants to get pregnant. In vitro fertilization, IVF. So, yes, IVF. So there's a lot of testings that are being done now, especially in bro ones, maybe in Lagos. They will do a lot of testing. So some women can even ask for designer babies if they want. I'm just joking. But, there's a lot of genomic testing going on, even in these places. So please let's just sit down in the university, close our eyes and think of just promotion. Please, let's think beyond that. There's a lot we can contribute to our own healthcare. By the little studies that we do, they add up. You understand? You do your own study, you give a result. That result is going to be useful to somebody else who is also doing something else. So that is the challenge I'm throwing to us. We all can play a role in this genomics in modern healthcare. All right, so I see that there has been a group before that talked about using Nutri-Sickle for medicine. So there's evolution of medicine. Initially we had the traditional medicine. There are the long-established practices and treatments. Then we now have evidence-based medicine. That means that it relies on scientific evidence for you to be able to make clinical decisions. Now we are going to precision medicine, where your genome is going to help the clinician to decide what kind of treatment he's going to give. And I think we need to challenge ourselves to get into that bandwagon. It's very important. One size does not fit all. One size doesn't fit all. I remember even simple malaria drugs. It works for some people. It doesn't work for some people. But so you see somebody who takes one drug and says he has malaria resistance, he has parasite resistant malaria. It may necessarily not be the parasite that is resistant. It may be that he himself is not metabolizing the drugs well. So there are things we have to start thinking about now. You understand? So we have people like that. They may take 3 or 4 different drugs before they feel better. So it's not one size that fits everybody anymore. So we have to start thinking of this precision medicine. What is it really about? It's an approach about disease treatment and prevention that considers your individual variation in your genes, in your environment, in lifestyle. And now they are even categorizing it into personalized medicine, stratified according to a group, tailored medicine, maybe according to your, So then P4, predictive, they predict your risk. They see how can we prevent it. They personalize it. Then you are even participating in your own treatment yourself. So there's a lot going on in the world about precision medicine. So this slide, I want to show in this slide that you say a large group of people and you are giving to them. In that group, some people in red that have serious side effects. How many people have seen that before with Fancida? Have you ever seen the side effect of Fancida before? I saw one time, it was terrible. It's as if the person had burns on his body. And that's because of the sulfur in the Fancida. So, for different people, and that's about 1 in 100. You understand? So, some people will have serious side effects. Some people will have no response. And maybe majority will have desired response. And we will say the drug is working. For those other categories of people, it's not working, right? So we need to begin to think about the genetic variation is. So we now know something that is called pharmacogenomics. So we have pharmacology on one side, the effectiveness of the drug metabolism, the side effects, and then on the other side, you have genes that have been inherited, you have the environment, what we now consider as pharmacogenomics. So with pharmacogenomics, we need to understand differences in the genes and how the body responds and the body's response to these medications. Does that person metabolize the drug or not? It's a combination of pharmacology and genomics to be able to understand genetic variations. And that's why Professor Lomola said that in answering the question they gave to him, he can't just answer by himself. There's also the genetics aspect that we have to consider. So even if we develop drugs, we now have to start thinking of not just only the side effects, we now have to start thinking about the genetic variants. Will this person with this kind of genetic makeup be able to metabolize this drug well or not? So there's a lot to consider. And that's why we are all important in this, conversation. Everybody has one little thing to contribute into this conversation. So what is genomics role in precision medicine? Genomics, is central to identifying the genetic mutations that impact whether you are at risk of a disease or you can respond well to a treatment. So you have the gene-specific, you have the genomic-driven diagnosis. So those are the things, that is how we contribute to precision medicine. And then you can use biomarkers. There are different types of biomarkers that can be used, whether to track, I'm still going into that next slide, whether to track the disease or whatever. So those are the useful things that that's how genomics is very important in precision medicine. And then you'll be able to clear diagnosis, you'll be able to say, this drug targets this particular biomarker, for example, and will be a useful treatment for this particular disease. So these are the different biomarkers that we have. You can have a biomarker that monitors disease. And biomarkers can be lipids, they can be genes, they can be proteins. So once you identify that this particular biomarker is associated with a disease, then it can be useful. It can either be monitoring, it can be a predictive biomarker, it can predict how severe the disease is. So those are the little studies we can do, basic science, clinical people combining together. Let us see particular protein, is it associated with a disease or not? Or can we use this marker for diagnosis? Those are the different types of biomarkers. So, so in genomics, we now have many useful studies. We have GWAS, that's the Genome-Wide Association Study. Like I gave an example of how a Genome Association study confirmed that, B blood group is susceptible to severe malaria. You have millions of markers, and then when you find these different markers, you can then take them in your population and then do a study to say, they found that all over the world that this particular marker can track a particular disease. Fine, they've published their own results. But the truth is that biomarker may not be useful in our own population. So we need to be able to answer that question ourselves. Maybe it's a different marker that is useful. We need to be able to understand. Such studies where a particular gene that was found in Europe to have different variations, and in the population it doesn't for our own population. So we have whole genome sequencing now. There's a whole genome. maybe concerning, you have cases, you have control, you'll be able to see what particular variants are more associated with the disease. And then you have